What are the relationship between neurons in the medial temporal lobes and the conscious experience of concepts? Do they reflect the phenomenological seeing? Medial temporal lobe, or MTL for short, includes neurons that respond extremely selectively to a few concepts. Medial temporal lobe includes neurons that respond extremely selectively to a few concepts. Their responses generalize across different views of the same concept. And these response properties prompt us to reevaluate the relation between the neural activities in MT and B4, B8 that we looked at, and also ITFFA that we looked at so far, and its relation to conscious experience. Can they really explain how we experience the world? That's the question that leads to this, that is followed by this video. So let's first remember this particular figure. Here is the schematic size of the receptive field of the V1 that is very small, and then the V2 that is much larger, and then V4 that is much bigger, and then finally IT. Okay, so IT neurons are, if they are responding to the face, then it can respond when the face is located here or there, whatever. So it doesn't have much selectivity in terms of the location of the stimulus. Okay. And if in monkey physiology recording, if the monkeys are trained to sort of the report the particular types of the object, let's say in this case, monkey was trained to discriminate or recognize particular rotation of the rotated paper clips. So here, this is a rotation of minus 72 degrees. And then the same neuron, the bottom figure is the firing rate of the neurons, but even if the same object is presented, if the orientation or angle is slightly different, then it doesn't respond at all. Or if the orientation looks kind of similar, but is not coming from the same clip, then it doesn't also respond. So this kind of response selectivity is called the tuning, neural tuning. And the tuning properties of the neurons in the IT is very complex and very selective. So this one responds to particular object, particular angle. And there are also neurons found in IT that can respond to a particular face and a particular viewpoint. So here, this is there's a neuron that responded strongly when monkey was seeing this particular face in profile like this. But this same neuron, when they are shown with a different angle of the same face, like this way to that way, sometimes when the angle is very similar to the maximally responding one, like this or the other side, then it can respond relatively strongly. But it doesn't respond when the face is presented in the, from the front or back and so on, the different angle. So it's kind of tuned to the face angle, okay? And furthermore, the same neuron responds to different person, but in the same kind of angle here, right? But here, this is again different person, and then different angle, and then now it doesn't respond, but the same angle of the optimum face seems to respond a lot. So there is something going on in terms of the identity of the face or property or feature of the face and the angle of the face. And this one is also interestingly the vast face kind of stimulus. And here, because of the background is black, the figure is depicted in black. It can be easily seen as two faces, but here it's more seen as a buzz, and then there's no firing here. So that's monkey's IT neuron response in particular situation. And something similar has been also reported in the human patient. So these are the patients who are the epilepsy patients who cannot be treated with the drugs. And to minimize the epilepsy seizure, they needed to be surgically removed by the with a very minimum excision of the brain parts. And to minimize the damage, they are implanted with lots of electrodes and then identify the source of the epilepsy. And as a part of this screening kind of process, they participate sometimes in this kind of experiment. And here, in one of these experiments, they were shown with a bunch of many different kind of faces. And in this particular patient's particular neuron in the left posterior hippocampus, researcher found that one particular neuron responded to this figure of Jennifer Aniston. So during the period of 500 milliseconds, when so this dotted line here corresponds to 0 millisecond to 500 milliseconds or so, that particular neuron spiked like 5 times or so, each trial that they presented this in Jennifer Aniston. Okay, and to see whether this particular neuron responds to a particular feature of the Jennifer Aniston, researcher then presented this, the Jennifer Aniston's different kind of faces, views. And then it turned out that pretty much regardless of how she looks like, original one is this, but even when the face becomes very small and including the body parts or background becomes blue or different, again, shape and or smile and so on, different angle, as long as it is Jennifer Aniston, more or less firing rate of the neurons were increased. And furthermore, when she was shown with different kinds of the famous people, like this is Kobe Bryant and Shaquille O'Neal, and other a famous person that this particular patient really liked, didn't show much response. And the animals or Eiffel Tower and Sydney Opera House and so on didn't also make a response. Okay. So this seems to be the neuron that is responding only when the patient saw the Jennifer Aniston. However, interestingly, this particular neuron didn't respond when the Jennifer Aniston was together with the Brad Pitt, which was who was the husband of Jennifer Aniston at that time, before they were divorced. All right. So then the next example of this type of the neuron is this Halle-Berry neuron, so-called. So this particular neuron again responded to Halle-Berry, right? However, it's quite so like Jennifer Aniston neurons. Regardless of how she looked like with sunglasses or with different color of the hair, the neuron responded whenever a patient see the Halle-Berry. The striking thing about this particular neuron is that when the Halle-Berry was presented as a drawing, so it's a black and white, right? Or Catwoman. So that's the character in a particular movie, Batman and Catwoman. They also produce a response. And then again, like the Jennifer Aniston neurons, other types of the celebrity didn't generate any response, including this. They did some kind of hint of the response for this other black lady, Oprah Winfrey, but selectivity is quite striking. And the remarkable thing as well about this particular neuron is that the text of the Halle-Berry also generated a huge response, but not other type of the text here, right? So it seems like this particular neuron seemed to be responding to the concept of Halle-Berry. Okay. How does a neuron compute something like this is currently unknown, but that's what the researcher has found. And then another interesting example is this left anterior hippocampus neuron. So this one responded to the Sydney Opera House. So when she saw this, Sydney Opera House, or when they were shown with the text of the Sydney Opera, it generated lots of response. However, they also responded to something similar to Sydney Opera. It's not quite a Sydney Opera, but it's actually the Baha'i tempo, right? But it turns out that this patient didn't recognize Baha'i tempo. And when they saw this, images, most likely they recognize it as a Sydney Opera. And consistently with this idea, other landmarks or similar looking tower didn't produce much response, and including the concept of the Baha'i, because they didn't recognize this. And so it seems like this neuron again responded to the concept of the Sydney Opera rather than the looks of the Sydney Opera itself. Okay, and then they also did the experiment along this line, but now modulated the visibility during the backward masking experiment. So for example, here's the 5 different types of the images they use as a sort of the control. And then when the stimulus duration was relatively long, like 264 milliseconds, which is very easy to see as a stimulus. It's a brief flash of the stimulus, but it's easy to see. Then the neuron recorded from right entorhinal cortex responded to World Trade Center like this way. And then when they presented the same stimuli with a shorter duration, like 33 milliseconds, then patient didn't recognize anything. So that corresponds to this no response pretty much here. But if it's 66 milliseconds, then patients started to respond or recognize sometimes, oh, this is World Trade Center. And this blue mark, by the way, corresponds to the trials where they reported seeing, the correct images. And in the case of 33 milliseconds, he didn't recognize it in any trials. But here, half the trials he recognized, and the recognized trial generated the response, and so on for the 132 milliseconds. And then when they look at average response, it turned out that across many neurons, when the patient recognized versus not recognize the image, if it's a very long duration stimulus, the response was very clear for the recognized trial. And fewer trials also existed, but there are responses here up to 1.5. And on the other hand, when the duration was much shorter, sometimes they recognized that seeing the World Trade Center or other images. And then they responded, made a response, relatively reliable one, up to 1.5, which is similar to here, unseen trial, but longer duration. And then, this is relatively, half of the trials. And then in the fewer, the more number of the trials where subjects didn't recognize the image, they showed a much lower response, below normalized firing rate of 1. So that's a sort of a population response, average. In summary, neurons in human medial temporal lobe selectively respond to a few concepts or maybe a single concept, which we don't know yet. But the question here from a consciousness research point of view is that do they actually correspond to what we report or what we see? In the paradigm that they tested, the response can be reflecting, report rather than something that we don't report, but we still see. And also, Another interesting feature of these responses is that although the stimulus itself is presented for a long time, like 1 second sometimes, or 100 milliseconds, or 500 milliseconds, response tends to be transient. Initially, there is a huge response, but it dies out. So the question is that are single neurons responsible for conscious percept of seeing things or not? And if that's not the case, then what are responsible for our conscious percept? And another question is that is there a better paradigm for the NCC search? So here, for example, even in the case of the backward masking, to modulate the visibility of the stimulus, researcher had to use different kind of duration of the stimulus. And so ideally, it is better to have a same stimulus and then have a different kind of percept to find out the NCC. So that's what we are going to look at in the next video.